Hello, my name is Vijeta Bhatt, and I'm a technology expert in our licensing team at Lonza. Today, I'd like to introduce you to Lonza's platform approach for producing engineered extracellular vesicles. Before we get started, I'd like to bring your attention to our legal disclaimer shown here. Here's your agenda for this talk. I will start with an overview of EVs as a promising but early therapeutic modality. Following this, we are very excited to introduce Lonza's novel exosome engineering technology, the Excite platform technology, which will be tailored towards projects exploring the unique properties of EVs. I will then go over product pipeline using our technology and a brief look at some clinical data. And lastly, how the Lonza platform can help you accelerate your entry into the clinic. There are several reasons how EVs could be leveraged as nature's own drug delivery system, due to which we believe that EVs could be the next big therapeutic modality. First, EVs are immune silent, resulting in higher tolerability within the body, which could make them favorable to repeat dosing. Next, they could potentially be used to deliver a vast range of molecules. Also, EVs have the ability as nanoparticles to cross biological barriers and allow retention. And finally, certain types of EVs have specific tropism for certain types of tissues, and this mechanism could be leveraged for targeted delivery. Therefore, as natural nanocarriers for delivering a range of cargo, exosomes could be exploited, making them an ideal candidate to engineer for drug delivery. For all of these reasons, we like to think of EVs as the body's FedEx, making them an ideal technology to carry complex payloads. EVs can be employed as a delivery system in different ways. For example, EVs can be used naive, that is, purified from certain cell types such as MSCs or HEK cells, and used as therapeutics, making use of their regenerative properties. In other instances, EVs can be engineered to payload cargo onto their surface or have the cargo be luminally loaded. This concept was demonstrated by Kodiak Biosciences in a publication from 2021, where they identified two scaffold proteins, PTGFRN and BASP1, that are preferentially sorted into EVs and enable the high-density surface display or luminal loading of a wide range of molecules. In either case, this was made possible through the engineering of the cell line using plasmids. That forms the basis of our Excite EV exosome engineering platform. This platform is based on the NGEx cell engineering technology, which was established by Kodiak Biosciences. Kodiak later validated this technology in clinical trials. NGEx utilizes two proteins, PTGFRN or BASP1. They are used to load cargo either onto the surface or into the lumen of the exosomes. Some examples of cargo that can potentially be loaded onto the surface of exosomes include antibodies, cytokines, receptors, and tropism modifiers. Cargo which can be loaded into the lumen of exosomes include antigens, enzymes, and peptides, to name a few. How this works is that our plasmids can be engineered to enable the fusion of the expression target to our scaffold. These plasmids would then be transfected into the target cell line, resulting in the loading of the payload to the surface or the lumen of the exosomes. Our license offer will include the plasmids to facilitate EV loading and will be available with the VS piggyback transposase to facilitate transfection. We are currently looking for partners to evaluate this exciting technology with us and welcome opportunities for beta testing. In the previous slide, we saw how the NGEX cell engineering technology can be used to express therapeutic payload onto the surface or the lumen of the exosomes. However, in case of smaller molecules, other tools and methods can be used. Oligonucleotides, peptides, or proteins can be linked to the surface of exosomes with high efficiency. We have protocols as well as IP in place to assist developers along those lines. The NGEX technology has been used to generate a deep pipeline of engineered exosomes aimed at treating a broad range of disease. This technology forms the basis for some of the clinical assets that Kodiak Biosciences developed. Some examples of these are ExoASOStat6, ExoSting, and ExoIL-12 that are shown on the right. These are good examples of how the NGEX cell engineering and post-purification loading technologies can be used to develop products that could eventually go into the clinic. Also here, we see that Kodiak has completed phase 1 studies with all of these assets. The examples we saw in previous slides suggest that engineered EVs can provide promising therapeutic benefits. Here, we have a case study where one of the patients was dosed with ExoIL-12 that was developed based on the NGEX technology. This therapeutic candidate is constituted of exosomes which express IL-12 on their surface via PTGFRN and is aimed to stimulate an anti-tumor immune response by activating T cells and NK cells. IL-12 is an important cytokine that can activate CD8 cytotoxic immune response and reactivate the immune system, typically with high cytotoxicity. However, here, Kodiak observed very specific tumor retention of the IL-12 molecules with no systemic inflammatory response. In the image on the right, we see 2 lesions that after 3 weeks of treatment with ExoIL-12 were responding well to the drug. Kodiak observed partial to complete response in a few patients with cutaneous T-cell lymphoma, suggesting that the treatment was safe and effective. I'd also like to briefly touch upon another potential application for engineered EVs using the ExoVac platform technology. ExoVac is a proprietary vaccine development platform that could leverage the unique properties of exosomes to deliver antigens and adjuvants simultaneously and selectively to the same antigen-presenting cells. Using the NGEX engineering technology, exosomes can be engineered to overexpress therapeutic molecules via surface display using PTGFRN. Adjuvants can also be incorporated with the antigen within a single exosome complex, boosting the immune response for infections. Using this platform could be advantageous for several reasons, such as selective targeting of the APC cells, a long-lasting immune response, thermal stability of the product, scalability of the manufacturing process, and most importantly, safety and tolerability. For these reasons, we can envision EVs as a powerful alternative for vaccines to other currently available options. So far, we have seen that EVs can be a promising therapeutic modality. But what key challenges do innovators face when developing EV therapies? Well, as we know, this is a new field and therefore takes some work to decode the biology and generate proof of concept data. Setting up the process and analytics can be challenging without the right expertise. Even with the right processes in place, additional experience is required to ensure scalability and GMP readiness. And further regulatory challenges are also anticipated as the product takes off. At Lonza, we have the IP, proven expertise, and a built-out ecosystem to address these challenges, having prior experience in the biologics as well as cell and gene spaces. With this holistic approach, we are looking for partners to bring these therapies out to the clinic. Our Excite platform technology is available to license as part of our Lonza in Your Lab approach. Lonza in Your Lab covers our platform licensing offers that allow you to evaluate and use our technology with our support in your own lab. Our licensing terms are flexible, both at the research stage and the commercial stages. The important thing to note about our Lonza in Your Lab approach is that the platforms we out-license to you are the same platforms we use in-house for our own CDMO manufacturing business. Our research license covers all preclinical work and allows you to create a number of cell lines and strains depending on your license terms. We offer a complete package including access to hosts, vectors, media, and an extensive know-how. I would like to end by discussing why you might consider Lonza to partner with. Having executed a number of development and commercial efforts, Lonza is a leader with experience in cell and gene therapy manufacturing across a range of modalities, and our platforms have been validated successfully in several projects. Our vision for the EV industry is to drive its advancement by bringing next-generation processes to the clinic. And to that end, we look forward to working with partners who would like to evaluate the potential of this promising novel technology with us. Finally, I would like to acknowledge our teams working on this platform. For any questions, please get in touch with us at licensing@lonza.com or scan the QR code for further information. Thank you.